For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
Okay, well, instead of going in circles, did you want to, we've touched on a lot of good points here. It's been fun. We're now at an hour and 35 minutes, but did you want to go into, is there any other points you wanted to make in genetics, or is there any last things you wanted to say, or something you wanted to touch on? I mean, I know I've said many times that the function of our DNA and the DNA element and the markers especially, we would predict that those are highly functional if the hierarchical patterns are there for design purposes. So I've talked to many evolutionists that'll admit, yeah, the majority of the genome should be based on evolutionary left. Have you read the Grar paper where he tries to make the case that the genome is no more than 20–25% functional, or else we would have gone extinct?
Yeah.
Yeah. Oh, yeah, we've talked about that before.
Yeah, and so I have a couple questions. Maybe I can state a few of my grievances, like maybe in a closing or something, but I'll try and get to a couple of them while I have you. So for kind of in reference to what you just said, if every genome was needed to be mostly functional, that doesn't quite makes sense to me for a couple of reasons. I mean, one, that paper makes sense, but it's talking about functionality in a very specific sense, in that it's a coding gene. There are obviously functions like
it does refer to. I feel like that's a mistake you often make. No, they're considering so. For example, in the non-coding regions, what some have called junk. Yeah, they've always expected some regulatory functions.
Yeah.
Some functions in the retrotransposons, the pseudogenes, some though. They've never predicted any more, just like with Grar said, any more than 20–25%. So you need to understand, and I'm 100% certain on this, especially after talking with like Frello and Mays, Conspiracy Cats. This includes non-coding and protein coding, because only, is it 2% of our genome codes for proteins. The rest of it, which is the non-protein coding DNA, that's doing other jobs. And I would say that the non-protein coding RNAs are regulating virtually all aspects of the gene expression pathway. But the evolutionist like Grar, he would disagree with what I'm saying. So there really are two separate expectations and predictions here. So no, he's considering the non-coding regions too, Snake. I mean, would you disagree with that?
That's the upper limit to the functional fraction paper, functional fraction of the human genome.
Yeah, because remember, he's the one that said if ENCODE is right, evolution is wrong. But ENCODE is what revealed through biochemical activity, right? They defined function as specific biochemical activity. They're the ones that said over 80% of our genome is active and transcribed into RNA. Well, RNA, DNA, RNA, protein, of course, not every time is the end product a protein. Oftentimes it just goes DNA transcribed into RNA, and then those RNAs have a variety of jobs. So that was the surprise there. So yeah, he's considering the non-protein coding regions. That's the majority of the genome for the most part.
Well, they're just considering basically necessary functions. They define it as a capacity that has been shaped by and is maintained by natural selection.
Right. Like, well, I mean, conserved regions would be maintained by.
So there would be regions that would have a biochemical function, but are not selected for, basically.
Right.
So, but basically, I mean, the larger point I was going to bring up is,
Yeah. Take your time.
like, so there's this thing called the onion test where we look
Right.
at, some onions have twice the size of a genome as other onions, and so we know for a fact that at least about half of the genome of the larger genome onions, that's not all functional. And things like amoebas some amoebas have the largest genomes out of any organisms, period. So either it took God more, it takes more information for God to make an amoeba, a single-celled organism, than it takes for God to make a human. Either that, or a lot of the DNA in the amoeba is basically not used.
Well, here's my response to that. So, in good point, the onion problem is definitely a good point to make. Plant genomes, though, they are larger for adaptive purposes. So think about it, Snake. Plants can't move. Okay, they can't put on a jacket if it's cold out. Okay, so if you're looking at temperature fluctuations, pH changes, for example, acid rain, for example, you name it. Okay, so plants are they are constantly facing negative environmental changes. Okay, and it can't do anything, so it has to adapt quickly, or it dies. So therefore, even like an animal, if you look at certain animals that can adapt, they can move underground. For example, humans, we can go in our house, put a coat on. So they need bigger genomes for redundant purposes, backup redundancy. And here's my question to you. I find redundancy to be fascinating evidence for design, because natural selection, which you were talking about, how it preserves these conserved regions because they're important. Natural selection has to do so. Natural selection works in the short term. It doesn't work in the long term. Therefore, how do evolutionary processes like natural selection build those mechanisms, like DNA repair mechanisms, redundant based mechanisms, for example, that work in the long term? How do you account for that?
How do we account for DNA repair?
So, for example, redundancy. So my answer to the
I
larger
mean,
plant
that's
genomes is because they need more redundancy. But natural selection works in the short term, not the long term. Redundancy is
but that
beneficial for the long term. How does natural selection evolve that redundant mechanisms and DNA repair mechanisms? Go ahead.
that's one of the functions of so-called junk DNA. Is it's not junk in the sense that it's garbage. It's
How does it
just,
evolve that?
how did it evolve it?
Yeah, how does natural selection evolve that which is necessary and beneficial for the long term? Natural selection sees the short term. How does natural
Well,
selection building a spare tire essentially?
yeah. So it's in the theory, it would be stuff that's like kept in the background in order for future adaptations,
So it knew.
stuff that can be
It
assembled. So how does
knew.
that get selected for? Is, I mean, we've observed things like fruit flies and things expanding their genome, and it's basically uh, loss of features happens a lot, and so you don't need every gene that's in the genome. So you'll lose some of the genes, but it's not that it gets cut out because it doesn't intelligently know, okay, we don't need to use that gene anymore, we'll just get rid of it completely. It's just that it kind of gets shuffled to the side, it doesn't get used anymore, but the instructions are still there, and it might be turned off and it might be deregulated, but most of the stuff is still there. And so natural selection doesn't know to just take out all the stuff that's not being used at the time. So it's just the way it works.
Right, but it's like, so for example, when we build cars with a jack or a spare tire, which to an evolutionary biologist, they would assume that that spare tire is the result of an evolutionary leftover. Or in computer code, we redundancy is considered good design. Backup code is beneficial. And then we see the exact same thing in the DNA code. We see redundant mechanisms. We see a need for more redundancy in plants. So that solves the onion problem. And the evolutionist now has to concoct a story as to how natural selection, which, as I said, sees the short term, not the long term, could evolve such elements and mechanisms that are clearly built for backup and beneficial purposes. But this type of multi-dimensional codes and these multiple layers of mechanisms and elements and whatever complexity in the genome, it's exactly what we would expect. Like, for example, one thing that fascinates me is alternative splicing. The fact that a single protein region, okay, of DNA can actually give rise to hundreds and sometimes thousands of different proteins. I mean, that's another thing I'm baffled as to how evolutionary mechanisms like natural selection can evolve such a thing. I mean, how would you explain alternative splicing, for example?
Well, if the transcription factor just binds slightly down the line, then it's going to be transcribing different parts. And I mean, if you translate something into protein, it's probably going to do something. There are very few inert proteins. So if it has a beneficial function, it's going to be conserved. But I mean, I've always said that evolution isn't incompatible with intelligent design. It's just not compatible with like young Earth creationism. So God certainly, if he created life, certainly would have put in the capacity for junk DNA if it was in his design that things should be able to evolve. If it wasn't in his design that things should be able to evolve, I would think that he would give us less of a capacity to adapt and would restrict the genome to like strictly 100% just what is necessary. And then whatever happens to that, well, that's just part of the fall. That's degradation and genetic entropy.
Well, when you get into biblical theology, it would make sense through God's foreknowledge. He would know we would have fallen, and therefore he would have built our genetics with redundant and DNA repair related mechanisms. But like you said, yeah, less than 2% of the DNA codes for proteins, and those proteins are functional and even multifunctional. For example, the protein moonlighting phenomenon, which is what we're looking to in making predictions on these hierarchical patterns in like cytochrome C, these ATP synthase proteins, for example. But guess what? What's the rest doing? This is the big question, and this is where evolutionists have to account for all that. As mostly evolutionary leftovers. Yeah, some of it's involved in regulation, but the majority of it is just there. It's just neutral junk for the most part. Maybe a hidden reservoir for evolutionary advancement in the future, like you were explaining earlier. But it actually turns out that the rest of it, the other 98% of the genome, is a huge control panel. You've got millions and millions of switches that turn protein producing genes on or off. I always explain it like if you go to a theater and you're watching a live show, for example, you got all your actors on the stage. There's a lot going on behind the scenes, and it looks like that this 98% of the genome, that's exactly what this is. They're there for a purpose, there for a function, and this is a testable prediction that we've made based on the created heterozygosity hypothesis. So if the vast majority of these DNA elements, redundant mechanisms, the genetics really is mostly functional, that turns those patterns that we've been talking about for an hour and a half into design patterns. Evolution, for evolution to be true, descent with modification, the majority really does have to be based on genomic fossils, evolutionary leftovers. Well, you mentioned conserved regions. They were shocked that there were so many conserved regions. For example, even between like the mouse genome and the human genome, you have these conserved regions that, you're right, natural selection preserves these because they're functional. If deep time evolution was true, and we've separated from obviously common ancestors with something as far on the tree as like mice, why are there so many conserved regions? Why isn't it more of a scrambled mess?
Well, I mean, there are some conserved proteins, but oftentimes you'll see that, again, they follow the evolutionary pattern, whereas like cytochrome C, it creates the same protein, but the sequence is much different in worms whereas the sequence is exactly the same in humans and chimps. It makes the same protein, but we can see that it's been accumulating and following a lineage pattern rather than just an intelligent pattern. Whereas if humans and chimps have been evolving the same amount of time, we would expect to see the same amount of differences between each kind. I did have, I don't mean to like rush you or anything, but I did have a couple of questions.
Of course, yeah, of course. Let me just comment on the cytochrome C example, because yeah, those are considered housekeeping genes because they're necessary. They're absolutely required for the maintenance of cellular functions. For the if they're not there, the organism will die. The existence of the cell will not be there. So like you said, and yeah, that is getting into some of the stronger evidence for universal common descent. Those highly conserved mitochondrial DNA proteins like well, even the CO1 gene, where you can look at DNA barcoding, these all fall within nested hierarchical patterns. But the thing is, the difference is, like, for example, the cytochrome C protein, those ATP synthase proteins in humans and chimps are nearly identical, like you said. But if you go down to like the old world monkeys, go down to dogs, fish, for example, mice, those highly conserved mitochondrial DNA proteins, they're different enough where they reflect descent with modification. But the question is, are those differences in those sequences a reflection of descent with modification or reflection of design and function? And that's why I was explaining the protein moonlighting, these proteins that have multiple functions. It's like a multifunctional tool. You got 10 different functions on these tools. I mean, this is science fiction. So we're taking that prediction. Jensen took that prediction on nuclear DNA proteins that have multiple functions, and he's saying that those differences in, say, your cytochrome C protein also reflect function that is specific, not only multifunctional, but functional specific to the kind of organism, right? Like the secondary function for something like an ATP synthase protein will be different in say metabolic optimization or cell optimization in a human and a fish, and that'll reflect those patterns. But guess what? Right now they haven't done the experiments. So there's a future testable prediction that once we do experiments, maybe mutagenesis on some of these differences in those proteins, we'll see, are they multifunctional or are they just reflection of descent with modification? So I'm just saying it's good you brought that up, and that just brings me to another prediction that creationists are making. But if you had another couple questions, yeah, go ahead and maybe we'll start wrapping it up there, Snake.